Hello students, today's topic of discussion is receptor mediated endocytosis. In endocytosis, we have a plasma membrane which invaginates, pinches off and form a membrane limited vesicle. The same process happens in receptor mediated endocytosis. We have a plasma membrane containing a specific receptor which binds to its ligand. The plasma membrane containing the receptor and the ligand buds inward, pinches off, and it forms a membrane-bound vesicle which contains the receptor and the ligand. In this lecture, we will understand how low-density lipoprotein are internalized via receptor-mediated endocytosis. Mammalian lipoprotein fall under two different categories. We have high density lipoprotein and low density lipoprotein. The class we will consider here is only low density lipoprotein. Here is a model of low density lipoprotein which contains an amphipathic shell made up of phospholipid, cholesterol and protein. The inner core is hydrophobic, containing cholesterol esters, triglycerides or both, triglycerides or both. It contains only a single molecule of one type of apolipoprotein, that is apolipoprotein B, and this protein appears to wrap around outside the particle as a band of the protein. This apolipoprotein B plays a major role in the binding of LDL receptor to its ligand. So, to summarize the structure of LDL, we have the outer layer which is amphipathic, made up of phospholipid, cholesterol and protein. Then we have inner core which is hydrophobic. This figure shows the time course of event in receptor-mediated cellular LDL processing determined by pulse chase experiment, where we took a fixed concentration of iodine-labeled LDL. These experiments demonstrate the order of event, that is, first there is binding of LDL to the receptor. This is the cell surface and here we have a receptor. The LDL protein binds to it. Second, there is internalization of receptor as well as our ligand, because now the ligand is tightly binded to its receptor. Here the plasma membrane buds inward, pinches off and form a vesicle containing receptor and ligand. Then this vesicle moves to endosome and then to lysosome. In lysosome, the third step, that is degradation of LDL, happens and the receptor moves back to the cell surface. In this experiment, we have taken the human fibroblast cell, skin cells, which we incubate in a medium containing iodine labeled LDL for 2 hours. So we have taken iodine labeled LDL and we cultured human skin fibroblast cell for 4 degrees Celsius. Then we incubate these cells at 37 degrees Celsius, but now we have absence of iodine labeled LDL. So in the next step, we measure the amount of surface bound LDL which is bound to the human skin fibroblast. We also measured the amount of internalized LDL and then the degraded LDL. As you can see in the graph, there is very much concentration of the bound LDL initially. With the course of time, the concentration of bound LDL decreases and that of internalized LDL increases. And after which the degradation of the LDL gets started. The figure here shows the model for pH dependent binding of LDL particle by the LDL receptor. The pH plays a major role for binding of LDL to its receptor. At the cell surface where the pH is 7, the LDL molecule binds to LDL receptor via apoprotein. This ApoB protein binds to the ligand binding arm of the receptor. This ligand binding arm contains 7 repeats. Out of this, the R4 and R5 repeat appear to be crucial for LDL binding. The beta propeller domain contains a histidine residue. In the cytosolic domain, there is a 4 residue sequence which is crucial for internalization. The 4 residue motif is asparagine, proline, X can be any amino acid, and Y is tyrosine. This NPXY sorting signal is very important for internalization of the receptor. After internalization, the receptor finally moves to endosome. At pH 5, the histidine residue of the beta propeller domain becomes protonated and it binds to the ligand binding arm with high affinity, and thus the LDL particle is released from the ligand binding arm. The ligand binding arm contains negative charge which binds to high affinity with the protonated histidine residue in the beta propeller domain. Here it is negative charge and the beta propeller domain due to protonated histidine is positively charged, which cause the release of LDL particle. The cell surface LDL receptor binds to the LDL particle via ApoB protein. This ApoB protein is embedded in the phospholipid outer layer of the LDL particle. Note that this ApoB protein interacts with the ligand binding arm of the LDL receptor. Two major coat proteins are there, that is clathrin and AP2 complex. The interaction between the NPXY signal of the LDL receptor and this AP2 complex incorporates the receptor ligand complex into forming endocytic vesicle. Before formation of coated vesicle which contains clathrin and AP2 complex, the clathrin coated pits which contain LDL receptor and LDL molecule are pinched off from the plasma membrane via dynamin mediated mechanism. In this mechanism, the dynamin, here we have coated pit, here we have dynamin protein which hydrolyze GTP into GDP and leads to release of the vesicle from the membrane. This coated vesicle now releases its coat and become uncoated endocytic vesicle. This uncoated endocytic vesicle is called as early endosome, which fuses with the late endosome. Late endosome has pH 5. Note that at the plasma membrane the pH was 7, which was beneficial for the binding of LDL to its receptor. In late endosome under acidic pH, the beta propeller domain now has high affinity for the ligand binding arm because beta propeller domain has histidine residue in its protonated form which interacts with the negative charge of the ligand binding arm, and thus there is a release of LDL particle. This late endosome now fuses with the lysosome and the LDL is broken down into its constituent cholesterol and fatty acid, and the protein is broken down into its constituent amino acid by the lysozyme enzymes. The LDL receptor do not goes to the lysosome, instead it is recycled back to the cell surface. Note that there is a pH difference on the cell surface and endosome due to which there is a conformation change in the LDL receptor. Here the LDL receptor has bounded ligand binding domain to the beta propeller arm, but here there is a free ligand binding domain which can bound to another LDL protein for the next cycle.